There is an unofficial hierarchy in ocean content. At the top, sharks. Below them, whales. Then octopuses, then jellyfish, then the deep-sea exotics with bioluminescence and translucent bodies. Below all of them, so far down that they barely register, sponges. A sponge does not bite, it does not hunt, it does not glow, it does not have eyes or a face or a way to express pain. It sits on a rock and does nothing visible. It has been doing this for roughly 600 million years. It was doing this before the first fish had a jaw. What is a sponge? The question sounds simple. The answer is one of the oldest arguments in biology. For centuries, sponges were classified as plants. They do not move. They are attached to surfaces. They lack every feature that makes an animal recognizable as an animal. No nervous system, no muscles, no stomach, no circulatory system, no respiratory organs, no brain, no symmetry in most species. A sponge has no front and no back. No top that functions differently from the bottom in any way that a biologist could use to orient it. If you cut a sponge into pieces, each piece will reorganize itself into a new sponge. If you push a living sponge through a fine mesh, the individual cells on the other side will find each other and reassemble. The organism has no central coordination. No cell tells another cell what to do. They aggregate by chemical signal and build a body by consensus. This is not a metaphor. In 1907, H.V. Wilson demonstrated this at the University of North Carolina. He dissociated sponge cells by pressing a living sponge through silk cloth. The cells, separated and scattered, migrated back toward each other over hours, reformed into clusters, and eventually rebuilt a functioning sponge. No other animal does this. If you separate the cells of a fish, you get dead cells. If you separate the cells of a sponge, you get a sponge again. The organism is not the body. The organism is the agreement between cells. The way a sponge feeds is the way a building ventilates. Water enters through thousands of tiny pores in the outer wall. Inside, specialized cells called choanocytes line the internal chambers. Each choanocyte has a single flagellum, a tiny whip that beats in rhythm, creating a current. The current pulls water through the body. Bacteria, organic particles, and dissolved nutrients pass over the choanocytes and are absorbed. The filtered water exits through a large opening at the top called the osculum. A sponge the size of a football can filter several thousand liters of water per day. It does this without a heart, without blood vessels, without a pump. The cells beat, the water moves. That is the entire circulatory system. There is no digestion in the way any other animal performs it. Each cell feeds itself. There is no stomach to break down food and distribute nutrients. Each choanocyte captures its own particle, absorbs it, and processes it independently. The sponge is not an organism that contains cells. It is a colony of cells that behaves like an organism. The distinction is not academic. It is the reason sponges survived every catastrophe that killed animals with centralized systems. The skeleton of a sponge is one of the most structurally efficient designs in the natural world. In some species, the skeleton is made of spongin, a flexible protein similar to collagen. In others, it is made of spicules, tiny structural elements of calcium carbonate or silica. And in the class Hexactinellida, the glass sponges, the skeleton is made entirely of silica, fused glass, grown at ocean temperature in seawater without a furnace. The Venus's flower basket, Euplectella aspergillum, builds a lattice of silica fibers that is structurally identical to the diagrid pattern used in modern skyscraper engineering. The lattice distributes stress across the entire surface. It resists bending, torsion, and compression simultaneously. The fiber optic properties of the individual spicules are comparable to commercial glass fiber. but the sponge grows them at ambient temperature while industrial production requires over 1,000 degrees. Material scientists have published papers on this. The titles are careful and technical. The subtext is consistent. The sponge solved a problem that engineering still has not. The lattice does something else. The geometry of the mesh creates internal currents that amplify the natural water flow through the sponge. The skeleton is not just structural. It is functional. It increases feeding efficiency. The architecture is the organism. Sponges appeared in the fossil record during the Ediacaran period, roughly 600 million years ago. Possible chemical biomarkers in Canadian rocks push that date further, to perhaps 890 million years, though this is contested. Whether the number is 600 million or 890 million, the fact remains. Sponges were alive before the Cambrian explosion, the event that produced most modern animal body plans. They were alive before eyes existed, before muscles existed, before the first brain formed. They watched in the only way a sponge watches, by sitting still and filtering water. As every major body plan in the animal kingdom evolved, diversified, competed, and in many cases went extinct. 5 mass extinctions have occurred since sponges appeared. The Ordovician-Silurian extinction killed roughly 85% of marine species. Sponges survived. The late Devonian extinction collapsed reef ecosystems worldwide. Sponges survived. The Permian-Triassic extinction, the worst in Earth's history, killed roughly 96% of marine species. Sponges survived. The Triassic-Jurassic extinction cleared the way for dinosaurs. Sponges did not notice. The Cretaceous-Paleogene extinction killed the dinosaurs, the marine reptiles, the ammonites. Sponges filtered water the next day. The pattern is not coincidence. It is architecture. A mass extinction event typically destroys organisms that depend on complex food webs, stable temperatures, or specific ecological niches. Sponges depend on water and bacteria. Water did not disappear. Bacteria did not disappear. A sponge needs nothing that an extinction can take away. No prey species to collapse, no symbiotic partner to lose, no temperature range so narrow that a shift of 5 degrees breaks the system. The body plan is so minimal that there is almost nothing to disrupt. There is a living glass sponge reef in the Strait of Georgia off the coast of British Columbia. It was discovered in 1987. Glass sponge reefs were thought to have gone extinct in the Jurassic, roughly 150 million years ago. Their fossils are found in European limestone. When divers found living ones growing on the seabed off Canada, the response from the paleontological community was not excitement. It was disbelief. The reefs are up to 9,000 years old. They grow at rates measured in centimeters per century. They are the slowest growing reef structures known. This is where the definition of success becomes a problem. By every metric we use for animals, sponges are failures. They do not move. They do not think. They do not compete in any way we recognize. They have no strategy, no behavior, no response to predators beyond being unpleasant to eat. If success means complexity, sponges lost. If success means speed, awareness, adaptability, intelligence, sponges are at the bottom of every list. But if success means time, sponges are not at the bottom. They are alone at the top. Every complex organism that evolved after them has either gone extinct or is younger by hundreds of millions of years. Sharks are roughly 450 million years old. Insects, 400 million. Mammals, 200 million. Humans, 300,000. Sponges, 600 million. Minimum. Possibly longer. The animal that does nothing has outlasted everything that does something. There is a tendency in science communication to frame evolution as progress. Complexity increases over time. Brains get bigger, bodies get more efficient, systems get more integrated. Sponges sit outside this narrative and do not care. They are a reminder that evolution does not have a direction. It does not reward complexity. It does not punish simplicity. It filters. And the filter is time. What passes through the filter is what works. Sponges work. They were here before anything had a brain. They will almost certainly be here after the last brain stops thinking. The hierarchy that puts sharks at the top and sponges at the bottom is a hierarchy of spectacle. It measures what is interesting to watch. It says nothing about what survives 600 million years. No brain, no plan, still here.